Howdy, folks. Thanks for checking into Mr. Ulrich's Land of Biology. I, of course, am Mr. Ulrich. In this notes cast, we're going to be building on previous discussions of meiosis and gamete production and talk about when and where those things come together and the implications of the location of fertilization and development. Of course, now that we have talked about sperm production and egg production, it makes sense that the next thing we talk about is fertilization, although logically, I guess it would be ovulation and ejaculation, but we'll get to those things when we get to the plumbing section and we talk about the equipment needed for those activities. Um, Now, most of my students come to me knowing that fertilization is when sperm and egg get together. If we go way back in history, the Age of Enlightenment time, there is what's called the preformative theory or a homunculus theory, where there was like a little they used to think there was a little tiny person like balled up inside of each sperm cell, like the sperm had the fully formed person. And then once it got into the egg cell, it could incubate, gestate, and blossom into an individual. We know that sperm are actually quite a bit less than a fully formed person. In fact, all a sperm cell really is half of a nucleus with a little outboard motor, right, driving around. And here's a scanning electron micrograph of fertilization actually taking place. Interesting to note that there is only a few, tens of sperm, maybe 100. Even though per ejaculum there are millions and millions of sperm, and we'll get to that. Why are the numbers so low at this point? Now, to form the zygote is going to be just one sperm cell that gets in there. And often my students ask me, how can it be just one when all of them are bashing their little heads against that jelly layer? Um, well, as soon as the tip of the sperm gets through that vitelline envelope, a very rapid chemical reaction takes place throughout the whole vitelline layer, and it seals it up, and it's no longer susceptible to the action of other sperm. Does it ever happen? Yeah, probably it happens, but really, really, really rarely one sperm fertilizes that egg. What are the first three rules of real estate? Location, location, location. And where the sperm meet the egg certainly is going to have some deep implications for the parents, for the gametes, and for the offspring. We see examples of external fertilizers in fish and in amphibians. One of the characteristics of external fertilizers, is that they make lots and lots of gametes because the world is a dangerous place. And when you're releasing your gametes out into the wild, especially those eggs with the yolk sacs, delicious, delicious, yummy, yummy eggs, they're organisms that are going to be out there and going to eat them. Now, one of the other things that we're going to see is that it's going to require a liquid medium because those sperm cells are motile and they're going to be whipping their little flagella around. And so the eggs are typically laid in the water. And there, of course, are exceptions to all of these rules, but the eggs are laid in the water and the sperm swim to the eggs. They're characterized by little to no parental care because you're letting your gametes kind of drift with the water and away they go. And typically you have no idea who the parents are. Now, again, there are exceptions to this. There are some fish who take quite a bit of care of their young, keeping them in their mouths and everything. But generally speaking, we see less parental care in external fertilizers than we do internal fertilizers. Internal fertilizers, on the other hand, the female retains the egg long enough for the sperm and the egg to meet within the reproductive tract in the safety of the female reproductive tract. Now, internal fertilizers get a kind of a choice here, depending on what type of organism you are, of laying the egg, the fertilized egg, and allowing it to develop outside of the body, or retaining the egg and having it develop inside of the body. And this is going to have implications on the types of organisms that we see as internal fertilizers. Reptiles tend to lay lots of eggs because though fertilization takes place inside, development is outside of the body, and they tend to lay them and leave them. They bury their eggs or leave them somewhere and take off. And so they got to lay lots of eggs in order to make up for the lack of parental care. Now, birds are known for lots of parental care, sitting on their eggs and watching after their chicks after they hatch. And so they only have to lay fewer eggs. They put more energy into each individual. Mammals lay even fewer eggs. We retain them within our bodies, most of us, within our reproductive tracts for the entire duration of development, and therefore we have even fewer offspring than birds or reptiles do. External development is when that zygote develops outside of the parent. And if this is the result of external fertilization, then it's taking place in an aquatic environment, and you're going to get the development of an aquatic egg. If this is the result of internal fertilization, then, well, we have an added problem. Now the terrestrial egg has to deal with water retention and waste removal that the aquatic egg doesn't have to worry about. Still, they have to get their nourishment from somewhere, and so thankfully mom has packed a bag lunch for each little embryo in the form of a yolk sac for that embryo to get the raw materials that it needs to make more embryo from. Of course, being outside and being a bagged lunch, there are going to be organisms out there that want to steal that bagged lunch. And hey, eggs are delicious, right? It's going to be a lot easier to take notes on this if you have your own unlabeled diagram of a terrestrial egg. You can use the one that I included in Google Classroom. You can go to MrUlrichsLandofBiology.com and get the one from there, or you can just do a Google search for a unlabeled terrestrial egg diagram, and it'll probably work. Now, we need to think of a terrestrial egg as its own kind of self-contained baby gestational unit. It contains all of the materials needed for that baby to grow and all of the equipment needed to protect it and allow it to go through all the processes that it needs to go through. So in there, snuggled in the inside, there's the embryo. It is surrounded by the well-known amnion and the amniotic fluid that's going to keep that developing embryo from drying out and keep it a little bit from getting too jostled. Other embryonic, extra embryonic tissues, or membranes as we like to call these things, are the yolk and the yolk sac. We talked about this, the bag lunch. It's going to get smaller and smaller as the embryo develops and uses up that material. Of course, as it does all of that metabolism, it's going to be producing waste products. Some are just carbon dioxide, and that'll go away, but there are other waste products too, nitrogenous waste, and those will collect in this other sac called the allantois or allantois. It also has lots of blood vessels in it for gas exchange. Surrounding all of this is the albumin. You know this is the egg white, but we haven't cooked this one yet. So it's clear this is a protein. You actually make quite a bit of albumin yourself. And I do too. Helps us maintain our water balance in our bodies. But the albumin as a protein is a source of protein amino acids for the growing baby as well, and it's going to keep it from drying out and from sticking to the outside of it, to the shell. Underneath the shell is the shell is porous. It's going to allow for gases to get through, and right underneath that is the chorion. The chorion is the membrane. That's the one that when you take a boiled egg and you get the shell off, and if you get it off in one piece, it's because it's all stuck to that little membrane that's underneath. That's the chorion. It's going to keep all of the moisture inside, but allow for oxygen and carbon dioxide and gas exchange. And of course, here at the end is the airspace. That's going to allow for pressure differences as when the egg is laid. It's warm. If it's coming from a chicken, it's going to be the temperature of a chicken, which is pretty warm. I don't know what that is off the top of my head, but then it is going to cool off, and as it does that, it's going to contract, and that's where that airspace actually comes from. Internal developers retain the zygote within the reproductive tract, where it's nice and safe. But of course, this means they require the specialized organs of the reproductive tract for this to take place. And early stages of development are embryonic, or we refer to the baby as an embryo. Late stages, when it's more developed, it is a fetus. We're going to see some membranes, some extra embryonic membranes that we saw in the terrestrial egg, the same ones, and they do the same job. The amnion and the amniotic fluid, same thing, surrounds that developing fetus, developing embryo, keeps from drying out, a little shock absorbing going on. And there is a bagged lunch that's packed. You look in this diagram and you don't see it because it's not there. We don't need it at this stage. We only need our bagged lunch from conception, which takes place in the fallopian tube, all the way down till it makes it into the uterus. So just a matter of a few days is all that yolk sac needs to last. And then it implants within the uterus. That's coming up. Here we go. The egg implants in the uterus, where a placenta develops from both the baby and from the mommy. So the placenta has material, embryonic material, extra embryonic membranes in it, and it has uterine mommy membranes in it as well. This is where the actual exchange of material is going to take place, not the exchange of blood, but the exchange of material from baby. Waste products will diffuse from baby's blood into mommy's blood so that her excretory system can take care of it, and the other way as well. Nourishments will make it from mommy's blood into baby's blood, where it can be used. It's important to set this apart from the umbilical cord. The umbilical cord is not the source of transfer. It just connects the baby to the placenta. The placenta is a pretty amazing organ, so let's take a little closer look. It is where we're going to see that other extra embryonic membrane, the chorion. The chorion makes the barrier between mommy and baby blood. Mom's blood pumps into these chambers where it flows around these chorionic villi. They're called these like fingery projections that grow into the chambers, and it's right through that chorion that the material is exchanged. And here's a picture of a human placenta to give you an idea of the size. Of course, if we're going to talk about placental mammals, we have to give a little bit of time to our non-placental mammal cousins, the marsupials and monotremes. Marsupials are the pouch developers. Mom gives live birth. She gives birth to a really underdeveloped embryo that has to crawl. It's like a little worm. It has to crawl through the fur from her vagina into her pouch, where there's a nipple, and that's where it gets its nourishment from. Waste products are going to go right into the pouch. Ew, so mom's got to clean those out quite a bit. There's no implantation, there's no uterus, nothing like that. Now, monotremes are, like our friend Perry here, they're the egg-laying mammals. They're still mammals, even though they hatch from eggs. As babies, they're poikilotherms. They're cold, and then they slowly warm up. They're still mammals because mom produces milk. There's no nipple. It just comes out of a region of her skin and her abdomen, and the babies have to kind of lick the milk out of there. Well, that's enough for now. Thanks again for checking into Mr. Ulrich's Land of Biology. I am Mr. Ulrich, and if you have any questions, please feel free to drop me an email. Put a comment section down here in YouTube if you're watching that, or in Google Classroom, or on Edpuzzle, or however you're getting it. Other than that, I really hope to see everybody in class. Be safe, y'all.